DNA Biology Project. This project explains how genetic information flows from DNA to proteins and how this leads to observable traits. Structure of DNA. DNA is a double helix made of nucleotides. It contains 4 bases: adenine, thymine, cytosine, and guanine. These bases pair specifically where A pairs with T and C pairs with G. The structure allows DNA to store genetic information. Requirements covered structure of DNA. Structure of DNA. DNA is a double helix made of nucleotides. It contains 4 bases: adenine, thymine, cytosine, and guanine. These bases pair specifically where A pairs with T and C pairs with G. The structure allows DNA to store genetic information. Requirement covered the structure of DNA. DNA replication. DNA replication occurs in the nucleus before cell division. The DNA strand unzips with the help of helicase, and DNA polymerase builds new strands. This results in 2 identical DNA molecules in a semi-conservative process. Requirement covered how DNA replicates. DNA to RNA gene transcription. DNA is used as a template to produce mRNA in the nucleus. RNA polymerase builds the mRNA using base pairing where adenine pairs with uracil instead of thymine. In translation, the ribosome reads mRNA in codons and tRNA brings amino acids. These amino acids join together to form a protein. Requirement covered DNA, RNA, protein pathway. Protein trait. The sequence of DNA determines the amino acid sequence of a protein. The structure of the protein determines its function, and this function leads to observable traits. Requirement covered how this leads to observable traits. Mutation. Mutations are changes in DNA that can affect proteins. For example, in acute myeloid leukemia, a mutation causes nerve cell growth, showing how DNA changes can lead to disease. Bonus helps full marks. In conclusion, genetic information flows from DNA to proteins through transcription and translation, and this process determines traits in living organisms. Thank you.